Hey guys, welcome back. Today we are going to learn the chapter 2, cell structure and cell organization. So to understand the functions of the organelles and the non-organelles, we need to draw it out. So guys, if you have printed out my notes, please draw it on your notes horizontally. If you don't have my notes on your hand, maybe you can use a A4 paper to draw it out. So I'm going to draw it out here. So guys, first thing you guys need to do is randomly draw a shape to show the plasma membrane of the cell. Now guys, remember you need to randomly draw it because there's no perfect cell that is living in our body. So you guys no need to use ruler, no need to use any tools to help you to draw the shape, just use freehand. Alright, but for illustration purpose, I will just use the shape in the software to draw the cell membrane, okay, to make it look a little bit neat. So guys, the first thing you guys need to do is draw a circle on the left side of the cell. Left side on the cell. Remember, please use your free hand, okay, randomly draw a circle, not the perfect circle. There's no perfect nucleus in the cell. All right, so this is the nucleus. This is the nucleus. We are drawing a nucleus and organelle. In PT3, your science teacher told you that, okay, nucleus is just like a black dot in the cell. Am I right? It's just like a black dot in the cell. But your science teacher didn't tell you the whole story of the nucleus. Now, nucleus is far more complicated than you have ever think about it. So nucleus is actually not a black dot. It actually has a black dot inside the nucleus. So this black dot is the nucleolus of the nucleus. Nucleolus of the nucleus. So what is the functions of the nucleolus? Basically, it is the brain of the cell that it is able to control the cell activities. So this nucleolus is actually inside the cytoplasm of the nucleus. The cytoplasm of the nucleus is not called the cytoplasm. It is called the nucleoplasm. Nucleoplasm. Plasm means fluid. The fluid inside the nucleus, we call nucleoplasm. Nucleo means nuclear. Alright, so inside the nuclear, we have some DNA material, which is called the chromatin. So all these string-like structure, they are known as the chromatin. All our informations are being stored here. That's why nucleus has another function, which is called the contains genetic material. It contains the genetic material. What is genetic material? One of the example of the genetic material is DNA. So DNA is being stored inside the nucleus. So all these things, they are like inside. They are like being surrounded by the membranes of the nucleus. So the membranes of the nucleus looks like this. Okay, so what you guys can see here is this is the membranes of the nucleus, and we call it as a nuclear membrane. Okay, we call it as a nuclear membrane. But from what you can see here, the nuclear membrane consists of two layers. These two layers nuclear membrane, we call it as a double membranes. So nucleus is one of the double membranes. It's one of the organelle that consists of double membranes. Later, I'm going to show you another two organelle that has double membranes in our cell. Alright, so that's it for the nucleus. Let's continue. The next organelle that you guys need to know is actually the endoplasmic reticulum. The endoplasmic reticulum, you can draw it like this. It extends out from the nuclear membranes, where it's shaped like this. So guys, look at this structure. Some parts of this structure, it has a ball sticking on the surface of the organelle. And this structure is known as the rough endoplasmic reticulum. So this is known as the rough endoplasmic reticulum. Rough endoplasmic reticulum. Now, the other side, what we have here is this is the smooth endoplasmic reticulum. So guys, write down smooth endoplasmic reticulum. So what is the difference between the smooth endoplasmic reticulum and the rough endoplasmic reticulum? Jot down. So smooth and rough, the only difference is they have, for the rough endoplasmic reticulum, it has the ribosome sticking on the surface of the endoplasmic reticulum. So the red dot here, this one is actually the ribosome. This one is being known as the ribosome. But for the smooth endoplasmic reticulum, it has no ribosome. No ribosome. So you can actually see that rough endoplasmic reticulum is really rough, okay, compared to the smooth endoplasmic reticulum. It is obviously rough because it has the ribosome on the surface. So rough endoplasmic reticulum and smooth endoplasmic reticulum, both of them, they have actually a very big difference in terms of functions. Don't want to confuse you guys, that's why we will start with the rough endoplasmic reticulum first. So this rough endoplasmic reticulum, it has the function which can transport the protein that come from the ribosome. That's why to understand the function of rough endoplasmic reticulum, first we need to look into this organelle, which is the ribosome. So guys, let's zoom in to this ribosome. We have another organelle here. Let's draw another organelle here, which is called the ribosome. Ribosome. Ribosome is actually the red dots over here. The red dots over here, all right? So ribosome, it actually looks like this. It has a big unit and the small unit. The job of the ribosome is actually protein synthesis. Now guys, let me ask you a question. If I ask you to cook a curry chicken and you don't know how to cook it, what are you going to do? You go for YouTube, right, to look for the recipe. So the ribosome is doing the same thing. They will need to have the recipe in order to produce something. So this recipe is actually come from the nucleus. And nucleus, they will provide the recipe in terms of the DNA or mRNA. So let's look at this one. This is the DNA. This is the chromatin, the DNA of the nucleus. It will be converted. It will be converted into a recipe called mRNA. Why only mRNA? Because this is the language that the ribosome can read. So this mRNA will go out from the nucleus through the nuclear pore and then go to the ribosome. So this ribosome, it will read the information inside the mRNA. Let's move this diagram to here. So again, this ribosome will take in the mRNA. So this is the mRNA. So mRNA will going from the left side to the right side. And as the mRNA is going from the left side to the right side, this ribosome will read through it, get the informations, and produce the things called protein from here. Okay, let's draw 3 circles is enough. So it will produce the things called protein. That's why the functions of the ribosome is the site of protein synthesis. Site of protein synthesis. The site of protein synthesis. Once the protein is being produced, you need to send it to somewhere else, right? For example, like the Lazada seller. If you have the product, you produce the product already, you need to deliver to the buyer. Correct? Now you need to use the transport to deliver to the buyer. So who is the one to deliver the protein? So the protein is being delivered by its friends called the rough endoplasmic reticulum. So once the protein is being produced from the ribosome, this protein is going into the rough endoplasmic reticulum, and it will go through the rough endoplasmic reticulum and then come to the end of the rough endoplasmic reticulum, and then it start to bud off. Bud off, it means it will like make a bubbles to wrap on the protein that produced by the ribosome and then release from the rough endoplasmic reticulum. So right now the protein is inside the bubbles and ready to be transported to another place. So the rough endoplasmic reticulum, it has the functions. The function is to transport the protein synthesized by the ribosome. Alright, so this is the most important thing you guys need to know about the rough endoplasmic reticulum. So where it will be transported to. Now, right now, the protein is just the raw material. Sometimes the cell, they want to modify them into something else. You're having the chips, okay? I mean, the computer chips. So this computer chips is just the raw material. You still need to add in some other electronics components to modify them to become something that you want. Am I right? So this rough endoplasmic reticulum is going to send the protein to an organelle that looks like a Wi-Fi. Guys, this is the Wi-Fi structure. I always call it. We call it as a Golgi Apparatus. So this Wi-Fi structure, we call it as a Golgi apparatus, will receive the protein that come from the rough endoplasmic reticulum. So the first thing they will do is they will fuse with the Golgi apparatus and release the protein inside. Let's zoom in a little bit. Okay, it will be released into the Golgi apparatus to be modified. So this protein will go through the Golgi apparatus. So while going through the Golgi apparatus, the protein will be modified by the Golgi apparatus by adding in the carbohydrates and so on. So this modified protein now is able to function properly already. So Golgi apparatus, it has one function, modifications. Modify the protein. So this modified proteins now is able to function properly already. So this Golgi apparatus is going to pack them together into a bubbles, just like the rough endoplasmic reticulum did. So it's going to pack them into a bubbles in this way with the modified protein inside it. Can you see that? Okay, that's why Golgi apparatus has another function called package. Okay, pack. After you pack it, it will transport it out from the cell, out from the organelle to somewhere that needed this modified enzyme. It's going to be transported out from the cell, or they will just stay in the cell for other purposes. Alright, so we have modified, pack, as well as transportations of the enzyme. Okay, so that's it for the Golgi apparatus. Now, let's come back to the smooth endoplasmic reticulums. That's also one of the important organelle that can be found in our cell. Now remember, the difference between the rough endoplasmic reticulum and the smooth endoplasmic reticulum is the smooth endoplasmic reticulum, it has no ribosome. Right? It has no ribosome. So another function for the smooth endoplasmic reticulum is they are able to synthesize the lipid. What is the lipid that we are talking about here? For example, like fatty acid and glycerol. So guys, just for your information, fatty acid and glycerol basically is the fat monomer in our body, and it looks like this. We are going to learn this in chapter 4, so no need to so worry about this first. Just jot down, okay, the lipid including the fatty acid and glycerol. Now, not only fatty acid and glycerol, the lipids also involve the phospholipid. What is phospholipid? Phospholipid is one of the components in the plasma membranes. Now, we have another important lipid called cholesterol. Cholesterol basically it is the raw material that can make other stuff. For example, like hormone. Hormone like testosterone, hormone like estrogens, those are the good stuff that can be found in our body. Okay, for example, like testosterone. Alright, how the smooth endoplasmic reticulum produce all these lipids? At first, they will have the raw material. And like how the rough endoplasmic reticulum produce protein, this raw material will go through the smooth endoplasmic reticulums, and with the help of the enzymes, in the smooth endoplasmic reticulums, it will butt off. From the smooth endoplasmic reticulums, and now the lipid is being produced. It's inside bubbles like this. Okay, so this is how they produce the lipids. Now, there's another very important function that can be done by the smooth endoplasmic reticulum, which is the detoxification. Anything that you consume, for example, like drugs, for example, like alcohol, will be detoxified by the enzyme that can be found in the smooth endoplasmic reticulum. So these detoxifications, normally they need a lot of enzyme. And all these enzymes can be found in the liver. Okay, as you guys know, liver is the main organ that helps us to detoxify those drugs, alcohol, and toxin. That's why inside the liver, we'll have a lot of smooth endoplasmic reticulum. Alright, guys, these two are the most important functions of the smooth endoplasmic reticulum. Okay, so now the next organelle that you guys are going to learn about is the lysosome. Okay, so guys, draw the lysosome. Lysosome is very simple to draw. Just draw a circle, and with some dot inside, then it's good enough to indicate this is the lysosome already. Now guys, what is lysosome? Lysosome is actually a membrane-bound structure that consists a lot of enzyme inside the membranes. So you can see this is actually the lysosome, and what is inside is they call it as a hydrolytic enzyme. It's the enzyme that can use to break down the bacteria. Sometimes the enzyme is able to break down the structure like protein, nucleic acid, or even some lipids. So how the lysosome they break down the food? Maybe I can show you something here. So basically, this is our lysosome. This lysosome is going to fuse with those things they want to break down. So let's say this is the bacteria. It will fuse with a vacuole that consists of the bacteria. Then this lysosome is going to release into vacuole that consists of the bacteria, digest the bacteria inside the vacuole. So this is how the lysosome works. Now, not only that, the lysosome can also break down a lot of stuff. For example, like let's say the Golgi apparatus now is not functioning anymore, it's being damaged. Lysosome will be the one to destroy Golgi apparatus by using their hydrolytic enzyme. Now, not only that, some animals like tadpoles, they have the tail at first, right? Correct or not? They have the tails at first. So this tail, when they grow into an adult as a frog, the tail will just disappear. But how this tail is being disappeared? By using the lysosome. So the lysosome can digest the cell that's unneeded. The tail will slowly disappear because of the digestion of the lysosome. Okay, now one more example is actually our fingers. At first, with our fingers, every fingers, they are like sticking together one. So how come right now we have 5 fingers like separated nicely? Because of the lysosome. Lysosome is able to digest the tissue or the cell in between our fingers to make us have 5 fingers. Now we have these 5 fingers, we need to say thanks to the lysosome. Okay, so that's all for the lysosome. Now guys, let's move on to another non-organelle structure, which is the plasma membranes. So plasma membranes over here. Plasma membranes. Okay, last time in PD3 we call it as a cell membranes, but in biology we don't call it as a cell membrane, we call it as a plasma membranes. Okay, so as you guys know, this thing here, they are the plasma membrane, am I right? This thing here, they are the plasma membranes. But this plasma membranes is not in a straight line actually. When we zoom into this plasma membrane, this is something that you guys can see. Okay, this is something that you guys can see. It is made up of 2 layers of small little stigma. This. Okay, inside it has a big chunk of stuff, and then stigma again. Alright, so the structure in the middle, this big things is called the protein. But for the small little guys here, this thing is called the phospholipid. That's why the main component of the plasma membrane is the phospholipid plus the protein. One of the characteristics of the plasma membrane is they are semi-permeable, or in a textbook, they call it as a partially permeable. Partially permeable. What is the meaning of permeable? Permeable means they allow something to pass through. This is partially permeable. That means there is something that can pass through. There is something that can be passed through. Something that can pass through. Something cannot simply just get through it. Something that cannot just get through it. Okay, so that's why we call it a partially permeable. Now, this characteristic is very useful as it can control the substance in and out from the cell. It can control which one to come in, which one not to come in. That's why the plasma membrane, it has a function that is able to control movement of substances into and out of the cell. Okay, so this is the function of the plasma membrane. Now we are going to learn more about the plasma membrane in the next chapter. So next chapter is going to a big topic to talk about only plasma membrane. Okay, so plasma membrane is one of the very important things that we need to know here. Alright guys, I think almost done already. Now we have the last 2 organelles that we are going to learn now. Alright, so the next thing that we are going to learn is the mitochondrion. The mitochondrion, it looks like this, and we are going to draw the inner membranes of the mitochondrion in this structure. Okay, so this is how the mitochondrion looks like. You guys label mitochondrion. Now, as you guys know, mitochondrion is the powerhouse for the cell. That means they can produce the energy, correct? Not that generates the energy. That generates energy. The full name of the ATP is called adenosine triphosphate. So this guys here, the mitochondrion is able to produce adenosine triphosphate. So what is adenosine triphosphate? Adenosine is the A tri means three phosphate group. Three phosphate groups. So adenosine triphosphates, it looks like this. And we have so many bonds here. The energy is being stored in the bonds between the phosphate. So we have the energy that is being stored inside the bonds of the phosphate. So this is what the mitochondria produce. So how they actually adapt to produce all this energy, then we need to check out the structure of the mitochondrion already. Now first look at the membranes of the mitochondrions. These mitochondrions, they consist of two membranes, the outer membranes and the inner membranes. As you guys can see here, it consists of two membranes as well. That's why mitochondrion is one of the double membranes organelle. It's a double membranes organelle. Why they want to have double membranes? Because they want to form a structure which we call the cristae. Now, cristae, it is a structure that can increase the total surface area of the mitochondrions. Okay, guys, try to imagine. This is the basic concept of the biology. I try to draw it here. Okay, guys, let's look at this. We have two lines here. One line is like this. This is line A. And then we have another line. This is line B. Okay, assume the length from here to here is the same as the B1 from here to here. Now, can you please tell me which one is longer? Answer should be B, right? Yeah, so B is longer and they can provide larger total surface area. When the total surface area is larger, they can make the process even more efficient. So this is the theory of the biology. Next time when you guys see the structure like this, okay, the folded structure like this means that they want to increase the total surface area for certain reactions. So same goes to the mitochondrion. They want to increase the total surface area in order to increase the rate of cell respirations. Okay, so this is the functions of the cristae. Inside the mitochondrion, we do have some cytoplasm inside the mitochondrion as well. But we don't call it as a cytoplasm, we call it as a matrix. Okay, the matrix. Alright, so the cytoplasm is known as matrix. Okay, guys, I think that's all you guys need to know about the mitochondrions. So without further ado, let's move on to the last organelle, which is so important in cell divisions. We call it as a centrioles. Centrioles. Okay. There's one thing that you guys need to know about centriole is this centriole can be only found in the animal cell only. You cannot find the centrioles in the plant cell. Okay, remember that. Now, centriole, it looks like a chopstick. This chopstick, they have the 9x3 structure. So they have 9 sets of microtubules, and each set consists of 3 microtubules. One, two, three, four, 1, 2, 3, 4, 5, 6, 7, 8. Oh, one more. Okay, so 9 sets of microtubules. We call it a 9x3 structure. Now, is this important? Not really important. Just want to show you only. Alright. So this centriole, it has a function to produce the spindle fiber. They are able to produce the form spindle fibers. Spindle fibers is a very important structure that's very useful in the cell divisions. Okay, so the line, the string like this, okay, they are able to form a string-like structure like this. This thing is called a spindle fiber. Okay, why they want to use it? Why they want to form the spindle fiber? For cell divisions. Cell division means one cell split into two cells split into 4 cells. So this is called the cell divisions that we are going to learn in chapter 6. Okay, so this spindle fiber, no worries guys, we will learn the spindle fibers again in the chapter 6, mitosis and meiosis. Alright. Okay guys, that's it for the whole map of the cell organelles for the generalized animal cell. So this is most of the cell components that can be found in an animal cell. Now guys, one big thing to take away, one tips to take away when you come to memorize the structure, the functions of the cell components. Try not to do it in a 3D form. If you go and Google search, you will see a lot of images that are in 3D. They draw the cell in 3D, they draw the organelles in 3D as well. So that 3D is not good for memorizing. If you want to learn the structure, please draw it in 2D like what we do right now. If you have done copying, then it's good enough already. Just for one time, you should be able to memorize the functions and most of the name of the organelle.